Assalamu alaikum warahmatullahi wabarakatuh. Manahil Kharamdarani here, and the topic of my today's presentation is cytogenetic abbreviations and nomenclature. So let's move toward it. First of all, cytogenetics. Cytogenetics is a branch of genetics that studies the structure of DNA within the cell nucleus, and you can say that it traditionally refers to the study of chromosome by microscopy following the application of banding techniques and permitting identification of abnormalities of chromosome, so that chromosomal number, chromosomal loss or gain of chromosome, its material, genetic material, or its positional changes of chromosome. The normal human karyotype. So, a normal human karyotype, we all knew that it consists of 23 pairs of chromosomes, in which 22 are autosomes and 1 pair of sex chromosomes, which generally arranged in order from largest to smallest. And we know that a karyotype is an individual's collection of chromosomes. And the term also refers to the lab technique that produces an image of individual chromosome. And so karyotype can be used to visualize abnormalities in the chromosome, such as an incorrect number of chromosomes, deletions, insertions, translocations, etc. These all are mutation. And the International System of Human Cytogenetic Nomenclature (ISCN) was established in 1978. So next thing, the main landmark of each chromosome are the centromere, abbreviation CEN, and the arms of chromosome. It divided into two arms: P terminal, short arm, and Q terminal for the long arm. Next thing is satellite chromosome. Centromere is also called primary construction, and satellite chromosomes are also called secondary constructions. These can be construct that serve as identifying markers. In addition to centromere, one or more secondary constructions can be observed in sometimes at metaphase stage. So they form a knob-like structure. It is called satellite chromosome or secondary constructions. Now further, a chromosome is any subdivision of a chromosome along its length is called region of chromosome. And chromosomal region includes parts that are not part of the chromatin, and each region is further divided into bands and subbands. So now let's move towards bands and subbands. What is band? So chromosomal banding refers to the alternating light and dark regions along the length of chromosome produced after staining with a dye, and you can say that black and white region, and it's not normal. It's usually observed after the staining of chromosome. And a band is defined as the part of the chromosome that is clearly distinguishable from its adjacent segment by appearing darker or lighter with the use of one or more banding techniques like Q banding, Jimsa banding, Gnaw banding, etc. And to clarify more the study of chromosomes, subbands, which are subdivisions of bands, these are used. So, for example, the CFTR genes chromosomal location is 7q31.2. It means that chromosomal number is 7. Q means the long arm, 3 means the bands, 3 means the region. It is the region 3, and it's the chromosome number 7. And 1 means the band 1, and 2 means its subband number 2. Point 2 means the subband number 2. If we say that cytogenetic bands, then these are labeled as P1, P2, P3, etc. And these subbands are also numbered from the centromere, from the centromere towards telomere, like P1, P2, P3, and Q1, Q2, Q3, etc. How to write chromosome? First of all, match the homologous chromosome according to their size. 3 things interfere in it: deciding ascending order, centromeric position, and banding patterns. These 3 indicators will serve as a guide for properly matching the chromosome, creating your karyotypes. So first thing is the right total number of chromosomes for the test subjects, like human have 23 pairs, dogs, pigs, etc. These things. So these are the chromosomal numbers. Next, place a comma behind the first number. This is followed by identification of sex chromosome from the total number of chromosome. 46XX mean 46 are the total number of chromosome and XX is female. 46XY mean that 46 total number of chromosome and XY is male. Then what is the use of plus or minus sign? So add a plus or minus sign to the effective chromosome number. It means that it is a mutation. It is anything that is not normal. So it is placing behind comma after the sex chromosome. First of all, we indicate that the normal sets of the normal chromosome number, then its sex of chromosome, and then plus minus mean that it's a deletion or insertion of anything. For example, Down syndrome have 21 extra set of 21 chromosome. It means that it have trisomy. And for a male with an extra number 19 chromosome, the karyotype would be displayed as 47XY. 47 means that the extra set 47. XY mean it is a female, it is a male. and 19 mean that it have extra 19 chromosome, additional chromosome. So next is cytogenetic nomenclature. Cytogenetic nomenclature is basically in its abbreviations, meaning, examples, and condition. So we know that 46XX is normal female karyotype and 46XY is normal male karyotype. And sen means centromere, del means deletion, 46XX, I already told you about 5P. It is female with Cri-du-Chat syndrome. Cri-du-Chat means that the infant cry like sound of cat due to deletion of part of short arm of chromosome number 5. Next is DAR. DAR means derivative chromosome. DAR 1. This translocation. Translocation means exchange of chromosomal material or genetic material derived from chromosomal number 1 and containing the centromere of chromosome 1. Dicentric chromosome. Dicentric chromosome, we already know that dicentric chromosome are the chromosome which have two centromere. So Dic XY means that the translocation chromosome contains centromere from the box X and Y chromosome, two dual chromosomes. Now dup means duplication, that extra genetic material. And Fra means FRA, fragile site. Fragile site means site at which chromosome have gap, constriction or led to breakage, etc. 46 total number of chromosome Y, fragile, mean that male with fragile X chromosome at the region X to Q to 37.3, mean Q and band 27 and subband is 3. Next is isochromosome. Isochromosome 46, example is XQ is that female with isochromosome long arm of the X chromosome XQ. Next is INV, inversion. Inversion 3P25Q21. It's a pericentric inversion. Pericentric inversion is basically reverse inversion. So reverse inversion at chromosome number 3. Next is MAR. MAR means marker chromosome. Marker chromosomes are basically small fragments of chromosome which generally can't be displayed without any special genetic analysis due to its small size. Then 46XX47 mean that extra set of chromosome. XX mean that it is female, and MAR mean that it is have a marker chromosome. That it means that a condition with a female will have extra chromosome and identified chromosome. Next is, time is very short, P. P means short arm of chromosome, and PAT. MAT and PAT. MAT and MAT are two terminologies. MAT means maternal region. 47XY dar 1 MAT. MAT is the condition in which there is an extra set of maternal chromosome, or there is loss of any paternal chromosome. So condition is that 47XY mean 47 additional part XY, but it is still male and additional part of inherited from his mother, so it is MAT, and vice versa paternal origin. Q means long half chromosome. R means ring chromosome. Ring chromosome are basically those chromosomes and chromosome of same size and length fused together, they form a ring. So they are called ring chromosome. It means 46XRX means female with the ring X chromosome. Then next is translocation. Translocation is basically exchange of chromosomal or genetic material. Reciprocal translocation, inverse translocation, and Robertsonian translocation is translocation between same things. Translocation between same things is called reciprocal translocation. So Robertsonian translocation, it is that acro fuse with acro, acrocentric telofuse with telocentric. This is called Robertsonian translocation. Then terminus 46, the original set of chromosome. XX mean it female. Q P ter means partial deletion of the long arm from the XQ21 to XQ ter. This sign is basically means detail is further given. So plus mean gain of 47XX plus 1, that female trisomy 21, it is a general case of Down syndrome. And then minus means the loss of 45XX minus 14 minus 21. It is normal female. 45 means chromosome is something is missing. And XX it means female. That minus 14 chromosome number 14 and chromosome number 21. Two chromosomes are affecting here. And karyotype is missing. Normal 14 and normal 21. And arms which are affecting are Q arms. Next is this sign. This sign shows that break. 5Q ter explanation is 5P15. It is deleted chromosome 5 in a patient with Cri-du-Chat syndrome, which I already told about you, with a deletion breakpoint in a band P15. Next thing is break and join. 2P terminal and then for the explanation given is 2Q21 break and join, 8p13 and 8p terminal. Description of derivative 2, portion of T2 translocation occur between 2 and 8 chromosome. Next is the mosaicism. So it is when a person has two or more genetically different sets in his or her body. It's called mosaicism. So 46XX or 47XX plus 8. It may be two conditions. Female with two populations of cell and normal karyotypes and one with trisomy 8. It can occur in both conditions. Time was short, so I presented very speedy. I hope that you understand all these things. For further queries, you can inbox me or you can email me. Thanks for your attention and thanks for watching my presentation. Jazakallah.